This next set of slides covers mechanisms of genetic variation. The first important concept here is that of a mutation, which is a stable, heritable change in the sequence of bases in DNA. The most common type are point mutations, which arise from the alteration of single nucleotides, but they can also be the result of addition or deletion of nucleotides. So in the first bullet point here, we're mainly focused on when a polymerase is laying down bases and puts the wrong one in a particular position, whereas here, something happens to the strand of DNA itself to cause slippage, for example, and results in addition or deletion of nucleotides, which I'll show you in an upcoming slide. Larger mutations are far less common. These are insertions or deletions of really big pieces of DNA or inversions of pre-existing DNA in which the hunks of DNA just simply flip in the genome. There's also duplication events that can take place in which hunks of DNA actually duplicate, and translocations in which pieces of DNA actually swap places in the genome. Mutations can also be spontaneous or induced. In spontaneous mutations, typically this is just a result of errors in DNA replication. So as I alluded to before, DNA polymerase III, for example, the main enzyme, has editing function and it can go back and repair things, but sometimes it doesn't happen. And so you end up with transitions or transversion type mutations. So a transition would be the alteration of bases, a similar basis. So a purine for a purine, or a pyrimidine for a pyrimidine. A transversion, on the other hand, would be, for example, a purine base being replaced by a pyrimidine base, or vice versa. These can also be due to insertion or deletion of nucleotides. They may also result from the action of mobile genetic elements like transposons, and so I'll post a brief animation of transposition. In this slide, you're looking at the concept of insertion and deletion, or indels. And in this sequence here, slippage leads to an insertion. And so here's our growing chain here, here's our template strand, and there's a slippage event, as you can see here. And the result of that slippage is to tuck this T into this tiny little loop here, and as a result, the daughter strand ends up getting an extra T. Over here, slippage leads to a deletion. So in this particular case, you can see the slippage resulting here in these two adenines being tucked away, and as a result, that pulls the template strand over a little bit, and the resulting daughter strand is missing two T's. Induced mutations can happen usually through chemical or physical agents. And so again, here's a couple bullet points on those and the types of them: DNA modifying agents that can alter a base, causing it to mispair, or intercalating agents that actually slide in between. They're usually planar type flat molecules that can slide themselves in between the DNA helix, or base analogs that are structurally similar to normal bases. We often use base analogs in antiviral medicines like azithromycin or AZT. So here's a list of examples of mutagens. And I'm not going to ask you to memorize these, except for I do want you to know the effects of the physical type, like UV light, which promotes pyrimidine dimer formation. So two adjacent pyrimidines in a nucleotide chain would form unusual bonds, and as a result, that kink would cause mutation. So it's it's the main reason why UV light is antibacterial, for example. Also, causes skin cancer. The effects of mutations are laid out here, and the first bullet point is basically just identifying the definition of the wild type, which is the most prevalent form of a gene. So a wild type gene that's found in the natural wild type organism for a particular species. A forward mutation is when the wild type exhibits a mutant form, whereas a reversion mutation is when the mutant phenotype actually reverts back to the wild type. Suppressor mutations are interesting in the sense they can be intragenic or extragenic, and I'll show you that in and a different slide. But the idea that microbes can sustain a mutation and then possibly alter the effects of that mutation with either a mutation with either an additional mutation within that gene or in some other gene is a pretty interesting phenomenon. And I'll give you an example of that here. So in this set of experiments, we've got two strains. Here's the wild type strain of Vibrio fischeri called ES114, and here's a mutant that my students and I made called C10. And C10 is defective in a cell wall amidase, and so this organism must produce this amidase in order to properly divide during cell division. One of the interesting things about C10 is that it's not motile, as you can see on the results of this motility auger. So here's ES114. Basically, you spot a little liquid culture on these soft auger plates, and the microbes will run out from the center if they can swim, which they obviously can't here. Here you're seeing the mutation, the mutant, with the gene added back, but the gene is not turned on in this particular strain, and so again, the microbe is not moving. Over here, you see the mutant with the gene added back, and the gene is then turned on with the addition of this chemical, and so the microbe can run again. What's interesting, though, when we get back to getting back to the concept of a suppressor mutation, is that if you let this spot sit long enough, you'll start to see these clouds of microbes running out from the center. And the hypothesis was that these are motile suppressor mutants, meaning that these guys have suddenly acquired the ability to swim, not necessarily through a alteration or fixing the amidase gene that was broken, but most likely through something else. And so the hypothesis was that it's probably an extragenic suppressor. And so when we used whole genome sequencing to ID the suppressor mutant gene, we found a particular point mutation. So this purple bar right here represents the wild type sequence. And here you can see at this particular position, base number 440, 441, 443 here is a T. But in all those mutants that are motile now, there's now an A in that position in this particular gene, and it's not the amidase gene. That was the interesting thing. So this is an example of how we would identify extragenic or even intragenic suppressor mutants. So this table highlights all these types of point mutations. First of all, the forward mutation. So this is none would be the wild type. And so you can see the wild type phenotype here and the resulting amino acid sequence that this wild type DNA encodes. In a silent mutation, there's a base substitution. So for example, ATG, ACA. So at this position here, instead of a C, there's an A. And that it's silent because it still encodes a threonine. Again, this is the third wobble position, and so ACA, that codon, and ACC both encode threonine. So the microbe doesn't even notice the damage. In a missense, there's actually a different base laid down. So instead of the C at this position, a G is laid down, and that causes the formation of a new amino acid at that particular position. In this case, instead of a serine, it's a cysteine. In a nonsense mutation, the change in one of the bases results in the formation of a stop codon, so a premature stop codon, and therefore this amino acid sequence is truncated, and usually that's a lot more detrimental. And then lastly, a frame shift in which there's some kind of insertion or deletion, and as a result, in this particular case, you see an insertion that throws the frame off, and so everything downstream of that frame shift is a completely different amino acid, most likely. In reverse mutations, here's the wild type sequence encoding methionine, threonine, serine. The forward mutation is a change in the DNA that basically encodes a new amino acid at that particular position. A reversion of that, and in this particular case, we would call this event a true reversion, in which the base goes back Oh, I'm sorry, it's not a true reversion. In this particular case, it's a reverse mutation that ATGACC. Yes, so it is a true reversion. Sorry, I confused myself there. But basically what happens is ATG, ACC, TCC, it goes right back to the original sequence. We think that these true reversions are pretty rare, but clearly it can happen. In this particular case, you see a forward mutation resulting in the codon encoding a new amino acid at this position, and then a reversion of that mutation in which the serine is replaced. And again, this is not a true reversion in the sense that a different base changes in order to still encode that serine. In this particular case, there's a base substitution as well, but we end up with a nonpolar amino acid being exchanged for a polar amino acid, and the individual ends up being a pseudo wild type because the polar amino acid, even though it's different here, it's still polar, and so the resulting protein, because of the similar chemistry, may still have most of its function. And so these types of mutants, these pseudo wild types, they're technically mutants, but phenotypically they behave like wild type some of the time. Here's an example of a frame shift, insertion, deletion. And so again, the forward mutation, you see an insertion, and that changes the reading frame here, and as a result, you get these different amino acids. And then a suppressor mutation that basically undoes undoes this particular mutation, essentially, a deletion that restores the reading frame. Even though there's a mutation here, the rest of the bases are the same. And so this intragenic suppressor mutant might survive and behave just similar to wild type. So again, intragenic suppressor, in which there's changes that happen in the same gene to roughly restore the wild type. Whereas in an extragenic suppressor, as you see here, and as I showed you here, the organism sustains a mutation in a gene. Say, for example, it loses the ability to swim, and then some other mutation somewhere else in the genome restores that ability to swim. Other types of mutations include conditional mutations, which are expressed only under certain environmental conditions. And so sometimes these are, for example, temperature sensitivity. So if you grow an organism at 35 all the time, you might never see this, but this conditional mutation might only display itself when you push the temperature higher or lower. And so you would never know it unless you played around with temperature. Sometimes the conditional mutations are only visualized when there's missing a certain nutrient, for example, which is the case here with auxotrophs. These are mutants rather, that are unable to make an essential macromolecule such as an amino acid or a nucleotide. So that's a conditional phenotype, and you wouldn't even know it. If you grow an auxotrophic organism on TSA or some nutrient-rich medium, the microbe might grow because it has all the nutrients that it needs. But if you grow that microbe on a medium that lacks that particular essential nutrient, and the microbe, because it's an auxotroph, can't make it, the microbe might not grow. The wild type strain from which an auxotroph arises is called a prototroph.